For this lecture, we will look at how eukaryotic cells break down glucose and create ATP molecules. In previous lectures, you learned that metabolism in a cell can occur because those reactions that release energy power the reactions that require energy. Exergonic reactions are linked to endergonic reactions. But the energy that is released must be harnessed, and a special molecule links endergonic and exergonic reactions. Can you guess which molecule this is? Yes, ATP. Remember, ATP is a special nucleotide. It not only contains adenine as its nitrogenous base and ribose as its pentose sugar, but 3 phosphate groups. The 2 phosphates furthest away from the ribose have unstable bonds. So when ATP is hydrolyzed, the bond between the last 2 phosphates is broken, resulting in a molecule of ADP with 2 phosphates and a free or inorganic phosphate, as it is no longer the functional group in an organic molecule. And as this is an exergonic reaction, energy is released, specifically 7.3 kilocalories per mole of ATP. Now, in metabolic reactions in the cell, the phosphate is not really free. After hydrolysis, it is transferred to another molecule along with the 7.3 kilocalories per mole of energy. The addition of a phosphate group to a molecule is called phosphorylation, and the addition of a phosphate alters the shape of the molecule being phosphorylated. In this example, the tyrosine kinase enzyme is activated when it is phosphorylated, and it can start catalyzing reactions. Take a look at this graph. Here's ATP. To form ATP, a phosphate group is added to ADP. And remember, ADP has a high amount of free energy. And here's the reactants, A and B. When reactant B is phosphorylated by ATP, it gains energy and is activated. This energy gain from ATP allows the reactants to reach the energy of activation, and the chemical reaction occurs. The chemical reaction results in A and B forming a bond, and the phosphate detaches from B. As this is an endergonic reaction for the reactants, the products have a higher free energy in comparison. ATP allowed this reaction to occur because it supplied the required energy. Cells make those ATP molecules during a process called cellular respiration. Here's the overall chemical reaction that occurs during cellular respiration. Glucose and oxygen are the reactants, while carbon dioxide and water are the products. This is an exergonic reaction, so energy is released, which is used to make those ATP molecules. As mentioned during the carbohydrates lecture, the nonpolar carbon-carbon and carbon-hydrogen bonds in glucose store a lot of potential energy. So when those bonds are broken, energy is released that can be used to make ATP molecules. And as the glucose molecule is broken down, the carbon atoms are released in the form of lower energy carbon dioxide molecules. But this overall chemical reaction is a little misleading, as there is not a single reaction occurring during cellular respiration. Instead, many chemical reactions occur, and many intermediate products are being created before all the final products have been formed. This is necessary, as the total amount of energy released from this exergonic reaction would cause the cell to burst into flames. Luckily, at each stage of cellular respiration, a small amount of energy is released that will eventually be used to produce ATP molecules. And it is important to remember that at each stage, enzymes are catalyzing these reactions. So exactly what types of chemical reactions are those enzymes catalyzing? A large proportion of the reactions are reduction-oxidation reactions, or redox reactions for short. These reactions involve the transfer of electrons. An atom or molecule loses one or more electrons, while another atom or molecule gains these electrons. You can remember this with a mnemonic device. Leo goes Gur. Leo stands for lose electrons, oxidation, while Gur stands for gain electrons, reduction. Let's look at an example of a redox reaction, the production of glucose. The carbon dioxide molecule is rearranged and the hydrogen atoms are added so that electrons are pulled more closely to the carbon atoms and they are reduced. The water molecule loses the hydrogens and the electrons are pulled away from the atoms, so they are oxidized. But in reactions involving organic molecules, the easiest way to determine where the electrons are going usually involves watching the hydrogens. How many hydrogen atoms does the carbon dioxide have? Yes, none. So the carbon atoms in the 6 carbon dioxides share no bonds with hydrogen. How many hydrogen atoms does the water molecule have? Yes, 2. So the oxygen atoms in the 6 water molecules share bonds with 12 hydrogens. Now to the product side. How many hydrogen atoms does glucose have? Yes, it has 12 hydrogens. So the carbons are now in a molecule that has 12 hydrogens. How many hydrogen atoms does the oxygen molecule have? Yes, the 6 oxygen molecules have no hydrogens. So which molecule gained the hydrogen atoms? Yes, the carbon dioxide molecules were reduced to a glucose molecule. And who lost the hydrogens? Yes, the water molecules were oxidized to oxygen molecules. Since cellular respiration is the reverse of the reaction we just looked at, will glucose be reduced or oxidized as it is broken down? Yes, glucose is oxidized to carbon dioxide during cellular respiration. Cellular respiration has 4 main stages. The first is glycolysis. The second is the processing of pyruvate molecules. The third is the citric acid cycle. And the fourth involves the electron transport chain and a process called chemiosmosis. Here's an overview of cellular respiration. What is the molecule directly going into the process? Yes, glucose. And which molecules are our important outputs? Yes, ATP molecules. We will answer this question in more detail at the end of the lecture. Let's start with the first stage of cellular respiration, glycolysis. Glyco means sugar, while lysis means splitting. So the word glycolysis literally means the splitting of sugar. This process does not occur in any organelle, but takes place in the cytosol of the cell. There are 2 phases to glycolysis. The energy activation phase involves an investment of energy, while the energy harvesting phase will be a payoff from our investment. In the glucose activation phase, 2 ATP molecules are invested to phosphorylate the glucose molecule. When the first ATP molecule adds a phosphate, glucose becomes glucose 6-phosphate. A second ATP adds a phosphate, and we get fructose 1,6-bisphosphate. The 1 and 2 of this name mean that the phosphates are on the first and 6th carbons. The molecule is now unstable with those 2 high-energy phosphates added on. This leads to our second phase. In the energy harvesting phase, the fructose 1,6-bisphosphate molecule splits in 2, producing 2 3-carbon molecules, DHAP and G3P. The DHAP molecule is then converted into a G3P molecule. G3P is short for glyceraldehyde 3-phosphate. We now have 2 G3P molecules. G3P loses electrons and forms 2 electron-carrying molecules of NADH. Phosphates are added to the G3P molecules to form 1,3-bisphosphoglycerate. Next, 2 molecules of ADP are phosphorylated to make ATP, and phosphoglycerate is created. The 2 phosphoglycerate molecules rearrange to form phosphoenol pyruvate molecules. The phosphates from these molecules are used to make another 2 ATP molecules. At the end of glycolysis, we have 2 molecules of pyruvate. Here's the big picture. Glucose is energized by 2 ATP and broken in half, eventually forming 2 molecules of pyruvate. The release of energy from this yields 4 ATP and 2 NADH molecules. But since we invested 2 ATPs, we only have a net gain of 2 ATP along with the 2 NADH. The NADH molecule that is made is an electron carrier. When NAD receives electrons, it is reduced to NADH. This molecule will be used later on to produce more ATP molecules. The ATPs that were made in glycolysis were produced by substrate-level phosphorylation. This means that an enzyme grabbed onto a substrate molecule with a phosphate and a molecule of ADP. then catalyze the reaction to add the phosphate to ADP, making ATP. Now, since glycolysis does not need to go on forever, it is regulated. When excess ATP molecules are formed, they bind to the enzyme phosphofructokinase. This enzyme's job is to catalyze the reaction that turns fructose 6-phosphate into fructose 1,6-bisphosphate. When the enzyme is inhibited by the binding of ATP, glycolysis slows down.